So in this talk, I'll be presenting our work on developing a virtual reality tool for neuron tracing. So the task that we're trying to address is a critical bottleneck in the brain network reconstruction pipeline. So in this work, we worked closely with collaborators in the neuroscience department. And so if we look at their pipeline, it's kind of a typical pipeline for a connectomics laboratory. So they start by labeling the tissue using kind of viral vectors that will make the neurons fluorescent. They then clear these using a technique like clarity, which renders kind of the fat tissues and other aspects of the brain optically transparent. Next, they can image the tissue using a confocal or two-photon microscope. And finally, they manually trace on the image slices or on a volume representation of this tissue, or on the scan data. So here, kind of this faint yellow line following this kind of blurry white structure is a trace that they've been working on. And finally, after they've completed all of these blue boxes, they can do the analysis that they're interested in, which is actually aligning these traces in the context of other brain maps. And they can use this to reconstruct how different parts of the brain communicate and use this to kind of understand the fundamental connections of the brain. And so if we look at kind of the overall pipeline, most pieces of this, the blue boxes here, are either kind of automatic or don't require a lot of like manual human time besides the tracing. So in their lab, they actually have kind of postdocs, research scientists, and hired kind of trained undergraduates who spend a significant portion of time just tracing on these image slices to get them to actually be able to even do the analysis they're interested in. And so this can take maybe months for even just a few neurons. And so work has been done to try and automate this task, but it is difficult to do in practice due to a significant amount of noise and complexity in the data, where it's hard to tell if features are crossing or if two neurons are branching or kind of running in parallel with each other. And so this picture that I have is just a subset of the data that our collaborators are working with. And you can see kind of the level of noise, especially in their data, where they're interested in long-range connectivity of the brain. So they also image at a lower microscope resolution so that they can acquire larger scales of data. And so in their workflow, their interaction with kind of the data that they're working with is through this 2D tool called Neuralucida. And so they sit at a standard desktop and use a mouse and then work through the data with kind of clicking along the neuron and scrolling up and down the z-axis here. So if you're trying to trace this neuron and you're following it as it goes down, so this kind of dot at the middle of the screen is where it starts diving down in the kind of z-axis that they've imaged. It turns into a dot kind of midway, and then it starts running horizontal again. And this is very hard to follow with their tool, and a kind of a common mistake that this sort of diving behavior will be missed entirely. And even with a volume rendering tool, volume rendering has been added in some other neuroscience tools recently, it still is difficult to kind of perform what is essentially a 3D task through a 2D interface using a mouse. When you click on a screen pixel, a ray is sent through the scene, and at some point it hits some kind of voxels data that is maybe opaque enough to trigger as a click on the voxel, or maybe it's not. And so you end up kind of having sort of a difficult system to work with. And we see this as an opportunity for virtual reality, where using recent hardware, we're able to actually provide a full immersive 3D interface that you can work in. So I'll start with kind of an overview of the current state of consumer virtual reality, talk about the design and technical challenges specific to our tool, look at our evaluation with experts, and then think a bit about virtual reality's future in visualization in general. So recent systems like the HTC Vive and the Oculus Rift are now available as off-the-shelf consumer products. They're affordable. I think they're both now about $600. They're easy to set up and maintain, and they can provide an immersive VR experience. So you have this headset that you wear on your head. You have these, in each case, 2 hand controllers that are tracked in 3D in the space. And you have these lighthouses that track your position of the head and your hands as you move around kind of a room-sized physical environment, which is mapped directly to your virtual environment. And in this way, they're able to provide a direct 3D interaction and a 3D environment for you to kind of immerse yourself and work in which makes them uniquely well-suited, we think, to performing tasks that require direct 3D interaction. So to give some sense of kind of what this might look like in a different sense, I drew the VIS17 logo in Tilt Brush, which is a painting application for VR from Google. So here I reach out with my right hand, which kind of is a paintbrush, and draw the parts of the circle. And then on my other hand, I have kind of this painter's palette, so I can change my paintbrush. And so the reality of developing tools for virtual reality is that there are a significant number of technical challenges to overcome to make your system usable in this environment. Because you're now responsible essentially for providing the user with their view of the world, there are hard requirements on your rendering performance and system latency. So rendering at 90 frames per second is kind of not an option at this point. You know, if you're dropping frames, the user will start to feel motion sick, and people can't use your tool for very long. As far as system responsiveness, when we work with tools on a desktop, we're used to kind of find your data set, click load, it hangs the whole UI for about 5 minutes, and then the system becomes responsive again. And this is also not really something you can do in VR, of just kind of freezing the user's experience of the world while you go do some processing in the background. Furthermore, user interfaces that we're used to from the desktop environment don't really translate. So if we look at kind of Neurolucida, which is the tool they use, or ParaView, which I'm sure most of us are familiar with, if you imagine kind of trying to work with this in VR, it doesn't translate very well at all. So in this work, we set out to design a new virtual reality tool for neuron tracing. And our design goals in the end are that this should be applicable for use for hours on end in their laboratory, kind of as a core piece of their workflow. So we worked with the neuroscientists who are co-authors and collaborators on this paper, and observed their workflow and kind of distilled down the key operations that we need to support in our tool. Another kind of limitation when moving to VR is that you have kind of a reduced input scheme in the form of the limited number of buttons that you have, and you don't really want to overwhelm the user with a lot of complexity in the tool. So we've reduced this down to just, of course, tracing neurons. And then additionally, we need the ability to navigate the data. So we display kind of a sub-region and allow the user to pan around, which we'll talk about in just a second. So the tracing neurons is implemented as a 3D painting system, where it works similar to Tilt Brush, where you essentially have this kind of brush controller. So here we show this little tip that is where your kind of trace will be painted from. And to trace, you just reach into the data and put your hand inside the neuron and hold the trigger and follow the structure as you trace. And we found this was really intuitive to use. It's also important to be able to trace the branches of the neurons, so to reconstruct kind of the full connections of where this kind of neuron cell is branching and connecting and talking to throughout the rest of the brain. And so this kind of elbow that's coming off. So we implement this as a free-form graph editing system, where the user can just start a new branch anywhere on the tree and trace it out, or start other branches out in the space and reconnect them back. For navigating the data, we display a sub-region to kind of avoid clutter and also help us ensure that we can maintain 90 frames per second rendering performance. And so in the world, we show kind of this floor, which is this dark gray thing. And we have this mini-map, which is the little blue box and the orange box on the left. And we show the bounds of the data set, which is the larger blue box. So in order to pan the data set, we map just kind of a one-to-one gripping of the volume, where you hold the trigger on your other hand and move it, and the volume moves in a one-to-one motion that matches this. We kind of experimented with, for larger data sets, mapping this as kind of a 2-to-1 or a faster motion, and found it kind of uncomfortable to work with. So the actions can be performed in combination, and you can also move yourself in the virtual space. So this is a playback of one of the expert users that we worked with in the evaluation, tracing one of our evaluation data sets. So he's kind of repositioning himself to get a better view at the data, navigating as he needs to kind of look at it from other angles and tracing it carefully. So as I mentioned before, a key challenge that we encounter and kind of is encountered when working with virtual reality is just ensuring that you meet the performance requirements to provide an immersive experience. So the high frame rate requirements place very strict demands on our tool, and having low latency both in kind of responding to the user's head motions and users' interactions is crucial for maintaining immersion. And so for this, we kind of split up our system architecture into multiple threads. So we have this rendering thread, which is responsible for kind of critical actions like displaying the next frame. And then we have a set of kind of GPU paging, disk paging, and processing threads, which perform different tasks like paging data off disk as the user's panning, paging it onto the GPU as it comes into the focus region, and performing any extra background processing that we need. Furthermore, we strictly budget the work that is performed on the rendering thread, and this enables us to stay consistently at 90 frames per second. And so even kind of hitches and drops, if you start turning on the kind of VR reprojection, this feels okay if you're not kind of interacting with something, but as you start dropping to 45 frames per second and you're kind of tracing your hand in front of you, you'll see it actually kind of jittering. And so it doesn't work as well if you're trying to provide an immersive tool for people to use. So key to achieving this as the user's paging through, say, gigabytes to terabytes of microscopy data is being able to page data onto the GPU without blocking the system. And so in order to do this, we implemented a two-level caching system. So we have kind of a first level that pages off the disk into CPU RAM, and another that pages off the disk, or off the CPU RAM onto the GPU. And on the GPU side, we take advantage of the sparse 3D texture, which is essentially a form of virtual memory. And so to meet the frame budget and kind of restrict how much work we perform on the rendering thread, we restrict how many pages we actually will upload and display each frame. And we perform the other tasks and uploads asynchronously in the background. And kind of anecdotally during the evaluation, we asked the users how they felt about the paging system, and their response to us was essentially, what paging system? So as you're working at the edge of the data and you're panning kind of slowly as you're bringing new data to work with, it's paged on quickly enough that you don't even notice it, and we kind of page a buffer around the focus region as well. So now we'll look at how we evaluated the usability of our tool. So we worked with 4 experts and 3 novices in our co-authors and collaborators lab, and had them trace multiple neurons in 2 different data sets over different 2 to 3 hour sessions in both tools. And this amounts to about 10 to 11 hours in the VR tool and in the Neurolucida session. And during the sessions in VR, people were free to kind of take breaks or resume, rest and stuff. We found that people tended to just that they would just consistently go through and work the 2 to 3 hours and felt comfortable kind of being in the environment for a long period of time, which I think is kind of an important testament to being able to provide an immersive tool that people can actually use in the lab. So here we have a picture on the left of the desktop software, and on the right of one of our co-authors tracing some neuron data in the lab. We collected feedback through informal discussions and a survey, and evaluated the tracing quality using diadem scores, which is a method in neuroscience for comparing traces of the same neuron against a reference tracing of sorts. So it would be scaled or scored kind of on how identical your trace is. So a score of 1.0 would be your trace is completely identical, and a score of 0 would be that your trace is completely dissimilar and not useful for analysis. So in our evaluation, we found that experts and novices found the VR tool more intuitive. So performing actions like tracing the data, navigating the space were found to be easier to do in VR. Things like creating branches, correcting small mistakes using the undo system was all found to be either a little bit easier or quite a bit easier to do, significantly easier. And they commented that the flexible 3D painting system allowed them to better focus on the data and kind of understanding the tasks that they're doing and seeing how the neuron structures are structured in the brain. They also noted that the immersive 3D interface improved their understanding of the data. So especially one of the novices commented that kind of after working in the VR tool and returning to do some of her sessions in Neurolucida, she was better able to understand the 3D structure of the neurons that she was actually tracing through the image slices. As far as kind of speed and quality, we find that experts have similar score distributions. So the green is the scores, a histogram of scores in Neurolucida, going from 0.2 to 0.8 scored on the diadem. So a score of 0.8 or above is an acceptable quality tracing for their analysis. And we find that on average, they're 1.7 times faster at tracing. In the case of novices, they perform better traces on average in VR and are 2.1 times faster and find the tool kind of easier to learn and more enjoyable to work in. So to wrap up, virtual reality we think is a promising tool for scientific visualization and potentially VIS in general. It can improve tasks involving direct 3D interaction and understanding 3D structure. So if your task that you're trying to do requires doing this, it could be worth looking at implementing in VR or exploring how kind of having an immersive 3D workspace could help. But many challenges remain to developing tools or porting existing tools into VR. So user interfaces and interactions that we're used to on the desktop must be redesigned. And the performance requirements is very demanding, especially for typical SciViz applications where 10 to 30 frames per second is considered quite interactive, which is fine on the desktop. In future work specific to our tool, we'd like to explore adding more guidance when tracing. In cases where experts made mistakes, we noticed that this could be attributed to missing a sense of the physical scale of the structures that they were seeing, or kind of forgetting certain things that the other tool would not let them forget, in a sense of when you create a branch point in our tool, it's sort of implicit. You're creating this new thing on the graph, and in Neurolucida, you explicitly kind of right-click and go through this menu interaction, and it will remind you to come back. So we can add additional guidance that will catch these common mistakes. And we also noticed that other mistakes were due to neuroscientists being unfamiliar with kind of SciViz representations like volume rendering or isosurfaces, and in other cases this led to them misinterpreting the underlying data that they were seeing due to just being unfamiliar with the representation that we were showing them. So thank you for your attention. My site has kind of the paper and the videos. You can email or tweet. And we'll also be giving demos of this during the poster sessions at the ski booth and potentially during the arts program. And we'd like to thank our co-authors and collaborators and, of course, the funding sources that supported this work. I'll take any questions.